Okay, we're going to talk about monosaccharides and disaccharides. These are all, you call these molecules sugars. They're all carbohydrates. But of course, carbohydrates would include big polymers like starch and cellulose, which we talked about before. We wouldn't really call them sugars. Okay, so let's have a look at monosaccharides. Glucose here, right? So this is well, the most important one, really. Oh, by the way, all of these molecules have the formula C6H12O6, glucose, galactose, and fructose. If you count up the atoms, you'll see that. So they are isomers of each other. Now, just to remind you that that structure there in blue, that's showing almost every bond in the glucose molecule. Remember that carbon always forms 4 bonds, hydrogen always forms 1, oxygen always forms 2. You don't have to remember the structure of it, but you just have to understand it. So usually you wouldn't write OH like that because it's easier to write just OH there. Likewise with that one. And this bit here you'd write as CH2OH, saves time and space. Also, we tend in the black structure above, you don't show all of the carbon atoms there, just for simplicity. And usually you have a little bit of shading here to show you that this edge is sort of, you're looking at this molecule edgeways on, and it's a flat six-membered ring, flattish six-membered ring with things, the H's and the OH sticking up and down. Okay, so that's the structure of glucose. You might remember that glucose exists in 2 forms. So I'm going to rub this bit out here. Glucose exists in 2 forms, alpha and beta. And the only difference between them is in the alpha and beta, this is alpha I've drawn here. In the beta form, you swap that H and the OH, they have opposite positions there. That's the only difference. Now, if you look at galactose, it is incredibly similar to glucose. The only difference really is that the H and the OH are in a different position to the H and the OH in glucose. Right, so that's the only difference between those two. They have different names. You might think, well, that bit's different as well, but that is just the alpha and beta forms. That can change in galactose as well. So this is actually beta galactose that we've drawn here. That's the more common version of it. Don't worry too much about that. But just so you get the idea that galactose and glucose, there's not a lot of difference in them. Whereas if you look at fructose, this is a little bit different because it's going to got a 5-membered ring, and it's got 2 carbons sticking off the ring, one there and one there. So it looks a little bit different. Now, we need to talk about tests for sugars. Okay, and the test that we use is, you probably come across it, is Benedict's test. Sorry. And with Benedict's test, you don't need to know the chemicals, but it's basically got copper sulfate in it, so it's blue to start off with. And if you have a reducing sugar present, it goes to a cloudy red color. Okay, it goes through various shades. It's got a bit yellowy, a bit orangey, and ends up being cloudy red. So you get a red precipitate, and that indicates that you have a reducing sugar in there. Now, that means it is a reducing agent, but don't worry too much about that. But it's just, if you want to know which ones Benedict's test can show off, right? It will react with glucose. Benedict's test, positive test with that. It will react with galactose, positive test with that. But it will give you a negative test with fructose. It will stay the same because fructose is said to be non-reducing. Okay, so now let's talk about disaccharides. And disaccharides are formed when you join 2 monosaccharides together. So we'll have a look at those. Right, so what I'm going to do is I'm going to form sucrose. So sucrose is the sugar that you put in your tea, and it's present in a lot of plants make sucrose, and during photosynthesis they initially make glucose, which is converted into sucrose before going was converted into starch first, and then converted into sucrose. So let's draw a disaccharide. So disaccharide means you've got two monosaccharide units together. So we'll get a glucose. Put that down there. And I'm going to get a fructose. And we're going to form a glycosidic linkage between these two. So I'm going to put them pretty close together, and I'm going to lose H2O from there. See, I'm going to lose, let's have that one back for later. Okay, I'll leave that out. Okay. All right, so we're going to lose an H2O molecule there. So if I just rub this out, so I'll keep one of the oxygens, lose H2O. And we've got that, which is sucrose. Now, sucrose is non-reducing. It will not react with Benedict's test, okay? Non-reducing, and the reason for that is, well, and you don't need to know this, but it is this bit of the glucose molecule which is essential for being reducing, and because that is now joined onto that oxygen in the glycosidic bond, that's the glycosidic bond, then it's not available to reduce anymore. So sucrose, non-reducing. Right, another disaccharide which you probably will have heard of is lactose. Okay, now lactose is found in milk, and it is made from a glucose and a galactose joined together. So if I get a glucose, that's where I don't think I want that bit of the glucose. I'll paste that over here. And I'm going to get a galactose. And we're going to form a glycosidic linkage between these two. I'll move that over to about there. Okay, and I'm going to lose water again from that. So you see I'm going to lose those H2O there, and we're going to form a glycosidic linkage. So there's the oxygen joined to that one. So that's a disaccharide that's called lactose. You've probably heard of lactose intolerance. Lactose is present in milk, which mammals are really only supposed to eat when they're babies. So most mammals, as they stop being, when they're weaned, their gut stops producing the enzyme lactase, which digests lactose back into the two monosaccharides. So the disaccharide lactose hangs around in the intestines, and it can't be absorbed, and then you get bacteria feeding on the lactose, and that causes like wind and diarrhea and stuff. So that's what lactose intolerance is. It's usually because you can't produce the enzyme lactase. Most adults can, but some lose the ability to do so. And the other one we'd have to talk about is a disaccharide of glucose. So and that is called maltose. The other disaccharides, maltose. You can probably guess where you find that from its name. So I'll just rub this out. I'll copy another glucose. And stick it next to it. Okay. And I'm going to move that over there. So those OHs come together to form the glycosidic linkage. Okay, we're going to lose H2O from there. We just got the oxygen in there. That's the glycosidic bond. Okay, that's maltose. And you get maltose You get it from the breakdown of starch. Okay, so you get it in your gut. Starch is broken down into maltose by the enzyme amylase, and then it can't get broken down into, you can't break it into, amylase can't break it into monosaccharides. So there's another enzyme called maltase on the surface of the intestinal cells, which breaks it down to glucose. You also get maltose in malt, and where you get malt from is you get barley, which is a seed. It's got loads of starch in it. You allow it to germinate, so you allow it to sprout, and the enzymes it will make will digest the starch into maltose for the plant to use for respiration. And then as soon as it germinates, then the plants, the little tiny seedlings are killed by heating them, and they're ground up, and they contain large amounts of this disaccharide maltose, which gives that characteristic malty taste. Okay, now maltose will give a positive test with Benedict's. Because the reducing bit, remember I said this carbon here has got to be like that. It's not involved in bonding, so that one is available to do the reducing. This carbon, this glucose, that wouldn't be available to do it because that's involved in forming the glycosidic bond. Okay. Now I've used this word hydrolysis quite a lot. Okay. Now what does hydrolysis mean? So I'll write it down here. Hydrolysis is the opposite of condensation, so the forming of a glycosidic bond. It's splitting with a water molecule with an H2O. So the way, let's say we break this glycosidic linkage by adding H2O to it. So what's going to happen is we are going to add an OH to this one. That's from the water. That's OH from the water. And that's the H from the water, so H2O. What have we done? We've split the glycosidic linkage. Now, how does hydrolysis occur? Well, hydrolysis can occur with enzymes can do that. That's what happens in nature. Or you can do it in the lab by you can heat with acid. Okay, so if you've got sucrose, well, it's now, I've hydrolyzed it now because it's now glucose and fructose again. But if you've got some sucrose and you boiled it up with acid for a while, you would end up with some glucose. That's this monomer here, glucose, and some fructose. And you can do an experiment with that. If you get fresh sucrose, test it with Benedict's, no reaction because it is not reducing. But then you heat it up with acid and hydrolyze it. It will react with Benedict's because now you've made some glucose, and that will give a positive test with Benedict's. Okay, while we're here, we need to talk about the iodine test for starch. Okay, so what is starch? Well, in the previous video, the starch is a polymer of alpha glucose. And of course, polymer beta glucose is cellulose. We can't digest that. We can digest starch. But what's the test for starch? You add iodine, which is iodine solution. So I2 in aqueous solution. Strictly speaking, it's iodine and potassium iodide, but don't really worry about that. And this is brown. And if you have starch there, it goes to a blue-black color. So you can use iodine to test for starch.